Hello everyone and welcome to our module on imprinting. Imprinting is an epigenetic phenomena. Epigenetics deals with variations in gene expression, not gene structure. So via imprinting there can be an alteration in gene expression and there can be differential expression of a gene depending on whether it came from the mother or the father. To understand this better, let's imagine that we have two copies of gene A found in the cells of a particular individual. One of those two copies came from the mother originally, and the other copy came from the father originally. Now, in most cells of our body, both copies of gene A can be expressed, assuming they're both functional and normal. However, for a small subset of genes, one of the two genes is said to be imprinted and therefore it is not expressed. So for example, if the maternal copy were not expressed, then only the father's copy of gene A would be expressed. And this phenomena is called imprinting. Imprinting occurs during gametogenesis before fertilization of the egg. Exactly how it occurs is incompletely understood, but genes are marked as being of parental or maternal origin. One of the ways they are often marked is by methylation of cytosine in DNA. At the bottom left side of the screen is the base cytosine, which you can find in nucleotides in our DNA. If it is methylated, meaning a methyl group is added, it becomes methylcytosine, and this is one of the ways genes can be modified sometimes in the process of imprinting. After conception, imprinting will control which gene is expressed. So in genes that are considered imprinted genes, only one allele is expressed, and this is in contrast, as I mentioned before, to non-imprinted genes in which both alleles are expressed. There are a small number of rare genetic syndromes that have been tied to the phenomena of imprinting, and the two classic disorders that involve imprinting are Prader-Willi syndrome and Angelman syndrome. What's interesting about these two disorders is that they both involve defects to the same segment of the same chromosome. However, you get a totally different condition depending on whether the maternal or paternal copy of the gene is abnormal. Both of these disorders involve chromosome 15, the section Q11 to Q13. So if you look at the bottom right side of the screen, chromosomes have a short arm, which is denoted with a P, and a long arm denoted Q. So the Prader-Willi syndrome and Angelman syndrome region of chromosome 15 involves the long arm, that's the Q arm, in the section from 11 to 13. If the paternal copy of this section of chromosome 15 is abnormal, the child will develop Prader-Willi syndrome. If the maternal copy is abnormal, the child will develop Angelman syndrome, and the differences are due to the phenomena of imprinting. To understand this better, at the bottom of this slide, I've drawn two copies of chromosome 15 in a particular individual. One copy comes from the father, that's this one here, and the other copy comes from the mother. On both of these genes, there is a section that contains PWS or Prader-Willi syndrome genes. However, these are normally expressed only on the paternal copy and not on the mother. And for that reason, I've written PWS here on the left side by the father's copy. There is also a section of genes called UBE3A, and these are normally expressed only on the maternal copy and not normally expressed on the paternal copy. For this reason, I've written UBE3A over here on the right by the maternal copy of chromosome 15. In Prader-Willi syndrome, there is a loss of function of the paternal copy of the PWS genes. You can just remember that the P in Prader-Willi syndrome goes with the word paternal. And as I told you before, the maternal genes in this region of chromosome 15 are not normally expressed. So when you lose the paternal copies, you therefore have no functioning genes in the PWS region for that particular child. About 75% of cases of Prader-Willi syndrome are from deletion of the paternal gene, usually due to a sporadic mutation. Another quarter are due to maternal uniparental disomy. I discussed this process in the video on meiosis, but in uniparental disomy, two copies of the gene are inherited from one parent. In this case, two copies of the maternal gene are inherited and no copies of the paternal gene, and therefore both copies are inactivated. Prader-Willi syndrome is the most common syndromic cause of obesity. This is a picture of a child with Prader-Willi syndrome. At birth, one of the first things that's often noticed is that these children have hypotonia. They can have feeding problems, a poor suck reflex. As they grow, they often have delayed milestones. They then develop hyperphagia and obesity. This begins in early childhood and persists through development. There's usually mild intellectual disability, and this is in contrast to Angelman syndrome, which we'll talk about in a minute, where there is severe intellectual disability. And then finally, these children often have hypogonadism and delayed puberty. Now let's talk about Angelman syndrome. In Angelman syndrome, there is an abnormal maternal chromosome 15 in the Q11 to Q13 region. This leads to lack of expression of UBE3A because, as I told you before, these genes are only expressed on the maternal copy, so when they are lost, you have no expression coming from the paternal copy. The majority of cases of Angelman syndrome are caused by deletions in the maternal copy of chromosome 15. A small percentage of cases are caused by uniparental disomy, and the reason this percentage is so small, much smaller than what is seen in Prader-Willi syndrome, is because you need paternal disomy in order to get Angelman syndrome. In other words, you need to inherit two copies of the gene from your father. Both genes will be inactive, and therefore you will develop Angelman syndrome from paternal disomy. In order for paternal disomy to occur, you need a meiotic non-disjunction event to occur in a male, and these events are much less common in males compared to females. In females, the process of meiosis is very drawn out, and therefore non-disjunction is more likely. The characteristic appearance of a child with Angelman syndrome is a child that exhibits frequent laughter and smiling. They are sometimes referred to as having a happy puppet appearance. 80% of children with this syndrome have a seizure disorder. They also often have ataxia and balance problems. And then finally, children with Angelman syndrome have a severe intellectual disability. And that concludes our module on imprinting.